goal in my research is to understand the molecular basis of the cognitive deficits, with the idea that if we understand the molecular basis of the cognitive deficits, we are in a better position to choose and to develop drugs that might help ameliorate these. And so what I'm going to be talking about really is the molecular basis of what is going on in the mouse models that we study. And that's different than the focus that many people take. So I probably don't have to do this to go over what Down syndrome is, but I feel compelled just for the sake of completeness. So it is an extra copy of the long arm of chromosome 21. Very rarely is Down syndrome caused by partial trisomy, and those partial trisomies have not informed us as to which genes are more likely to be relevant to Down syndrome, the phenotypic features of Down syndrome or not. So they haven't really helped. They're too rare and not well enough characterized phenotypically, more often than not well enough characterized molecularly. I don't talk about 21p, the short arm. From our annotation, there are approximately 500 genes on chromosome 21, and I'll come back to that a bit later. And the important thing is these genes are normal, they are not mutations. So what we are looking at here is simply an extra copy, elevated expression, largely, although there are specifics behind this overexpression. And this is just an overexpression of these genes. There's not a problem other than having too much. And I'll come back to this also, but we expect there to be perturbations because of the overexpression of these genes in many genes that are not mapping on chromosome 21 and many, many cellular processes. I also need to mention that there is no Down syndrome critical region. We need to think about genes all over the chromosome. Julie Kornberg showed when this was first proposed that one can be diagnosed with Down syndrome, including intellectual disability, and not being trisomic for the Down syndrome critical region. So I figured myself that that meant it was dead in the water, but it has persisted in the literature and shouldn't. So the phenotype of Down syndrome is certainly very complex, very variable among individuals, involves many organs and organ systems. But what I am interested in are the particular consistent phenotypic features. All individuals with Down syndrome have some level of intellectual disability, although that is variable, and they all develop the neuropathology of Alzheimer's disease at an early age, and the majority of them, if they live to be older than 60 years, will also develop a dementia similar to an Alzheimer's-like dementia. So the social consequences of Down syndrome, I think, are huge, which make this an important target for considerable research. And our interest is in pharmacotherapies, and Down syndrome is the most common genetic cause of intellectual disability. In the United States, the incidence remains at about 700, 750 per live birth. 1 in 750 live births. It's a little bit lower in Europe. And the estimates that I've seen are there more than 300,000 individuals with Down syndrome in the United States, making the idea of pharmacotherapeutics for adults with Down syndrome an important idea also. Thanks to many features, in particular better health care and integration into the community, the average life expectancy now of a person with Down syndrome is greater than 50 years and is probably going to increase. The average IQ is 40 to 50. This is a significant impairment. Yes, there are people who are at low normal IQ level, but there are also many who are more profoundly impaired. And the thought that has been in the Down syndrome basic research community for many years now is if there were methods for improving the IQ, maybe 10 to 20 points, you could shift a large proportion of individuals with Down syndrome from cognitively impaired into the region of low normal. And this would greatly add to their ability to participate in the community independently. So the neurological phenotype that we consider is the brain, the Down syndrome brain, is grossly normal at birth, whatever that means. I mean, this is what you will see in the literature. But it is clear, I think, that postnatal development of the Down syndrome brain slows, and as a result, there are smaller sizes of several brain regions, among them important regions like the hippocampus, which keeps coming up in Down syndrome research, decreased numbers of cells in various brain regions, and decreased numbers of particular kinds of cells. The complexity of the neuronal connections in dendritic arbors and spines is abnormal. And then there is, with age, an increasing loss of functional markers in the basal forebrain cholinergic neurons, which provide cholinergic input into the hippocampus. Spatial deficits in Down syndrome, possibly the most focus has been put, or not spatial deficits, cognitive deficits, possibly the most emphasis has been put on spatial learning, and that has been supported and possibly initiated from data in mouse models, which implicates the hippocampus, but probably all brain regions in the Down syndrome brain are relevant, are impaired, and have abnormalities in some facet. And this is what we tended to look at in mouse, was hippocampus and hippocampal function. However, these are other features that also need to be considered, whether we are looking at phenotypic features in people with Down syndrome or trying to make and analyze and evaluate mouse models of Down syndrome. People that have impaired hearing, vision, are going to have impairments in learning unless these are adequately corrected. Speech production is often difficult. If you can't communicate with people, it becomes less important what you know if people don't know that you know it. And another topic that several people are really beginning to give some attention to are sleep impairments, in particular sleep apnea, interrupted sleep, insufficient sleep, insufficient quality of speech, certainly now very clearly contributes to cognitive deficits. And I have heard some people say that if you could just fix the sleep problems, not only would the families be happier if the child was sleeping reliably through the night, but you would go a long way towards facilitating learning and memory. Incidences of seizures, autism, and depression as they become into adolescent years seem to hover, certainly with seizures and autism, around 10%. And then, as I already mentioned, the early onset of Alzheimer's disease. So I just want to show this slide. I mean, everybody here knows that if you talk about intellectual disability, not all intellectual disability is the same. So there are specific features. But this is a study that was published 20 years ago by Ursula Bellucci and her group, and they compared performance on certain tasks in people with Williams syndrome and people with Down syndrome. And there are two things that I think are interesting. So these are the typical developing kids. This was the task. We produce these figures, and here's what Down syndrome reproduces, here's what Williams syndrome reproduces. And the point here that I think is fascinating is not just that these are very different. The errors are very, very different between these two groups. But this is a task we ask children to do. How do we assay something like this task performance in a mouse? And it comes back, brings us to the question of when we use a mouse model, what we ask a mouse to do, how, even if it is assaying a hippocampus, how do we know that what we are asking the mouse to do, and when we correct the mouse's behavior, rescue the mouse's deficits in that task, how do we know that it is relevant to what we might be asking a person to do, a task such as this? So there are many problems, many challenges in Down syndrome research. So the outline of the presentation, what I wanted to do first was go over some background and give you the current status of the gene content of chromosome 21, which is work that my lab continues to do, to go over a little bit of the mouse models of Down syndrome, not all of them, and then importantly talk about the status of the preclinical evaluation of drugs using a single mouse model of Down syndrome with the idea of rescuing learning and memory deficits in people with Down syndrome, and then subsequently where we are as a result of this in clinical trials for Down syndrome. So I want to go over that first, and then I'm going to talk about the work that my lab is doing currently. We are focusing really at the level of protein expression, what is right, what is wrong, what changes, what doesn't change, protein expression and the perturbations seen in 3 different types of experiments, looking at learning and memory in the TIA65DN mouse model of Down syndrome, and then showing you the differences that we see and the similarities when we look at protein perturbations in 4 different mouse models, and then lastly, touch upon something which is fairly recently become extremely important gender differences. So the gene content of chromosome 21, my lab has spent some time over the years looking at the genomic sequence and using a number of different evaluation tools to determine what genes are there or what we predict might be genes. And the current number is we are around 500 genes and gene models. That doesn't mean we know much about what these genes do. Out of these, we know that there's about 160 that really code for proteins. We know this because we can see these proteins, people have studied these proteins, we find these proteins in multiple other vertebrate models, or even all the way down to yeast. So we're confident that there really are this number of protein coding genes. We're also confident that there are 5 microRNAs, and we are also confident that there are a large number of transcripts that we don't know whether they code for small proteins, large proteins, or are functional RNA molecules. When we look in mouse, we find that the protein coding genes that we're confident of are largely conserved in mouse, and so are the microRNAs. But these genes here that we don't know whether are protein coding or functional RNAs are not conserved at the sequence level. So there are a lot of these novel genes that we don't understand the functions of, but when we look at the sequence, which is really how we compare DNA between 2 organisms or RNA between 2 organisms, we don't see sequence conservation. This doesn't mean there is not functional conservation, because it's becoming increasingly clear that a functional RNA can be very, very short and degenerate. And that is very difficult, if not impossible, to find when you're looking over a few kbs of RNA sequence, never mind over kilobases of it. So that's where we are. That's what we know. The other thing that we know, and that has caused, is the reason for a lot of challenges in Down syndrome basic research, is that these genes that we understand that are conserved in mouse are distributed on 3 different mouse chromosomes. The telomeric region of mouse chromosome 16 has about 100, and then 2 internal segments, one on mouse 17 and one on mouse 10, respectively have about 20 protein coding genes and 40 protein coding genes. They also have, of course, mouse-specific genes. So the immediate thing that comes out of this, well, first I need to say again, the candidate genes that we need to think about are all the genes on chromosome 21. There is no logical way of saying, we don't need to look at this gene or those genes or this region. We don't have the data to support that. So an important feature that comes out of this, which is immediately apparent to anyone who wants to make a mouse model, is this is really difficult. If we want to make a good model of Down syndrome, we need trisomy of all 3 regions. And we don't have that, but I'll come back to that. So I want to mention a little bit about the chromosome 21 protein coding genes, because we can talk about those. If you look at the functional annotation of these genes, this is a summary of them, and I just want to mention that essentially all these genes are expressed in brain in some time, someplace. So we can't use that as a filter for cognitive deficits, even if we wanted to. But if you look at the functions here, there are 20 transcription factors, or genes that are known to function in modulation of transcription. There are another 10 that we know are involved in processing of RNA, processing of transcripts generated by transcription factors. And then there are a number of proteases and protease inhibitors, a number of proteins that function in modifying proteins, which means they regulate the activity and the localization of their targets. So methylases, kinases, inhibitors of phosphatases, there's even a sumorylation, and then there's a number of genes that function in the ubiquitin pathway, which regulates protein degradation. So if you overexpress all of these, you have to expect that expression levels at the RNA and the protein level of all sorts of things that are not on chromosome 21 are going to be affected directly by this or downstream. You overexpress a kinase, so you overphosphorylate the targets of that protein, and they go on to overdo or underdo whatever they normally do because of their excessive modification. So we have to expect perturbations throughout the genome and throughout the proteome. And the other thing that's important here is we can't understand Down syndrome by looking at the genes on chromosome 21 one at a time. We can learn what the genes on chromosome 21 do looking at them one at a time, but we don't know how they work together doing that to produce the phenotype of Down syndrome. So I just did this little schematic pointing out some interesting features that we associate with learning and memory. So learning and memory itself, dendritic spine abnormalities, neurodegeneration, histone modifications, and MECP2. Then these are, each one of these is a chromosome 21 encoded gene, and these are from single gene studies, and a number of these, when overexpressed, affect learning and memory. A number of them also may modify or affect histone modifications. We observed abnormalities in dendritic spines. So this is all individual genes. So the challenge here is, okay, I know what these genes do. What do they do together? And that's really a profoundly difficult, important, but difficult question to answer in Down syndrome. So back to the mouse model issue. So mouse as a model system, and through my conversations over the last 2 days, yes, I know that mice are not people. And ideally, we want to work with people. But mouse is our preclinical evaluation system, part of what should be a set of systems to do preclinical evaluation of drugs. And the big thing is you can assay learning and memory and behavior, okay? The limitations of these are the gene distribution, 3 different chromosomes, species-specific genes, and interactions among genes. So if I only look at genes on mouse chromosome 16, I don't know and don't have the consequences of interactions that they may have with genes encoded on other chromosomes. So it is only in the last few years that Eugene Yu up at Roswell used Krelok systems, chromosomal engineering, and created these 3 mice, which I will be calling the DP16, the DP17, and the DP10, DP standing for duplication. So he chromosomally engineered mice, 3 separate lines, each that are trisomic separately for one of the 3 regions, okay? So obviously, just breed them all together, okay? Anyone who breeds mice knows that there's nothing simple about this. So you breed 2 together, you get 1/4 of your offspring will have both DP10 and DP17. You breed these to the DP16 that are a little more fragile. They have heart defects, similar to seen in people with Down syndrome, so they don't all survive. So you end up, when you breed all these together, about 5% of your offspring will be a complete trisomy. In a typical behavioral experiment that we do with 1 drug, we need 40 of those mice. So you need a bigger mouse colony than we have. So then you have to make compromises, and every time you make a compromise with your mouse model, you have to be thinking about what that means for your extending or trying to extend your results to the human condition. So the most popular mouse model is the TS65DN, and this was constructed about 20 years ago by Muriel Davison at the Jackson Lab, and Muriel Davison is one of my very best friends. So this was the first viable model. It remains the most popular because it has been around for 20 years, not because it is currently the optimal model among nothing that is perfect. So here is a schematic of the TS65DN. Here's chromosome 21. Here's the entire 16 region that is present in the DP16 line, and here's the TS65DN. It has most, but not all, of the syntenic region on mouse 16. And Muriel said from the very beginning, it's the telomeric segment of mouse chromosome 16 translocated to the centromeric region of mouse chromosome 17. But in those days, before we did genomic sequencing, we look at a region and we say, oh, it's small, can't be important. Okay, it was indeed small, but it encodes actually 50 protein coding genes, which was shown by Jan Hero's group about 3 years ago, about 50 protein coding genes. These are not orthologs of genes on chromosome 21. They are not trisomic in any individual with Down syndrome. They are irrelevant. They are confounding, I think. You have to assume, okay? Your basic assumption is they are confounding. So it's trisomic for 88 out of 161, and it's trisomic for an extra 50. So it's not a perfect model. It does have great phenotypic features that are relevant to Down syndrome. Reduced size of hippocampus and cerebellum, reduced cell numbers, abnormalities in dendritic spines. It is impaired in hippocampal tasks, very robustly shown to be impaired in these kinds of tasks, also robustly been shown by many groups to have decreased LTP and enhanced LTD, loses functional markers, a lot of good phenotypic features, okay? But we have to keep in mind that the genetic basis is not completely orthologous to the genetic basis in Down syndrome. And that has to interject at least caution in our conclusions. So, TS65DN, been with us 20 years, okay? A lot of groups all over the world have studied this and found compelling Down syndrome phenotypic features. The TS65DN is the only mouse model that is being used in preclinical evaluation of potential pharmacotherapies. Okay, it's the only mouse model that has been used. So this is a partial summary of the drugs. I couldn't put them all on because I thought the print would get too small, but this is a good representative list. And each one of these drugs listed here, and here are its targets or its mechanism of action, what we know. Each one of these has been shown to rescue at least one learning and memory task in the TS65DN, okay? Some of these have only been tested with one task. Some have been tested with more than one and rescue more than one. Some have been tested with more than one and rescue only one, okay? So there's nothing perfect about any of these. But the interesting point in this slide, I think, is if you look at what they do, GABA A receptor antagonists and NMDA receptor antagonists, which act in opposition, inhibiting inhibitory neurotransmission and inhibiting excitatory neurotransmission. Now, you're not injecting these drugs with any specificity in the brain. You're giving them to the mice in water or milk, or you're injecting them. So these drugs are everywhere. They act on opposite features of neurotransmission, and yet they are both successful. Memantine, approved for clinical trials in Alzheimer's disease. So we have, you can read these as well as I can, Prozac, fluoxetine, works very well, minocycline, cholinesterase inhibitors, 2 of them do, but donepezil does not correct learning and memory deficits. Melatonin and lithium, a green tea component, agonist of sonic hedgehog, antioxidants, a whole variety of things. Now, I went to Drug Bank and I looked, and I don't find anything common, even in the off-target effects known of these drugs. There are little indications that there are some similarities, there are some overlaps perhaps in secondary targets, or what are considered certainly off-targets and maybe negative features. But there is nothing here when you look at this to say, aha, I understand why all these drugs work. We don't understand why these all work. However, everything with an asterisk, these here, memantine, EGCG, antioxidants, has been through clinical trials or is in the process of clinical trials now. Largely, things are not working out really well. Cholinesterase inhibitors, they did donepezil, and these have been very small trials, conflicting results, largely inconsistent, limited, not really exciting. Antioxidants was done in a very large trial. There was considered to be no positive outcome. And there are possibly very good reasons for that, because it was done in the TS65DN, and what we know about some of the genes are not trisomic in the TS65DN, they would have a very negative effect in an antioxidant environment, actually be neurotoxic. Memantine has been done in 2 trials, a large trial with older adults and no positive results, and a small trial with younger adults, and I think arguably no positive results. Similarly, the trial with EGCG, again, a smaller trial with younger adults, sort of 18 to 30-ish, again, arguably no positive results. A group in France treated infants 3 months to 3 years or 2 years with folate. And after they got the results, which were just all over the place, they found that the cohort in there, about 100 individuals that were also on thyroxine for hypothyroidism, showed and that were on folate did have positive outcomes, positive improvements. So they have immediately gone into a larger trial. Pentyline tetrazole, they are recruiting now in Australia, and when I talk to MDs about this, they all tell me that this is not going to be a good idea. Pentyline tetrazole used at higher doses than were used in the TS65DN induces seizures. And it is actually used experimentally as a seizure, to induce seizures. It is not approved by the FDA. Children with Down syndrome are at increased risk of seizures. So there are many in the medical profession who would say that pentyline tetrazole is not a good idea. And they are recruiting in Australia. And then Roche has a trial ongoing, and they have reported what they say are extremely exciting results in a trial that's been going on for about a year. They're still recruiting. So we'll see what happens there too. So the rest of my talk is really devoted to why are clinical trials failing, what do we know, and what do we need to know in order perhaps to do better clinical trials or more productive clinical trials. So one of the issues is the drugs that have been tested, with the exception of 2 of them, have no known connection to a function of chromosome 21. So we're not addressing a problem that we know how mechanistically it is caused in Down syndrome. We are addressing a feature. And these are features that are seen in many mouse models, certainly, and also seen in many causes of intellectual disability. We are modeling in mice, right? The TS65DN is a mouse. So we have the very fact that it's a species difference. And as I mentioned early on, when we ask a mouse to do something that is not necessarily the same complexity or addressing the same problems when we ask a child with Down syndrome to do the tasks we ask of a child with Down syndrome. And there's also the pervasive problem of strain differences. Each one of the Down syndrome mouse models is on its background, and that's what it's on. What would happen to the phenotypic features if you could move it to a different background? We don't know. And then, as I said, the TS65DN is a partial trisomy, and it has confounding effects of genes that aren't on chromosome 21. So to make the assumption that a positive drug response on a behavioral phenotype in the TS65DN can be translated to Down syndrome is making 2 serious errors. It is assuming that the 45% of chromosome 21 genes that are not in the TS65DN don't affect the phenotype and do not affect drug responses. And the same thing for the non-chromosome 21 orthologs. We also are assuming, or they are also assuming, that these genes do not create any of the features in the TS65DN and similarly are not influencing the drug responses. I think from what we know about these genes here and the genes that are missing, I think these are very dangerous assumptions to make. So, my lab started several years ago now with the idea of looking at protein profiling. RNA is not very predictive of, protein levels. So while you can do RNA, look at gene expression at the RNA level very accurately now, very rapidly, it doesn't tell you what goes on at the protein level. It doesn't tell you about the differences in levels in protein, and you cannot look at protein modification. And modification in proteins is what governs protein activity. So the observations that guide some of the work we do, we have this serious knowledge gap. We don't know much about what chromosome 21 genes do. We know some stuff, but we don't know what else they do, even of the best studied genes. And we don't know all of their interactions with each other. And so I don't think we cannot make, at this point, knowledge-based predictions of which genes are important to the cognitive deficits in Down syndrome. And so what we set out to do was to look at pathway perturbations. And for this, I don't need to know what genes do on chromosome 21. I need to know their effects, their consequences, how those consequences may be leading to learning and memory deficits, and how to control those consequences. So I want to step into a technical aside very briefly. Anyone here who has worked in protein measuring quantitative assessment of protein levels knows that Western blots are really tedious. They can be accurate, but you don't want to do that for too long. We are using a technique in my lab known as reverse phase protein arrays, and this is a schematic here. basically what you do is you do not take antibodies, you do not take RNAs. You simply take your protein lysates from your brain region or whatever you're interested in, and you spot these on a microscope slide that's been coated with nitrocellulose. Similar in concept to oligonucleotide arrays, where you array oligonucleotides or RNAs on a microscope slide. We are arraying proteins from the brains of our mouse models. And then you screen them with antibodies in a process really similar to what you would do in a Western blot. You screen with an antibody, and then you screen with some method of detecting where the antibody has bound. And that's what's showing here. So all these little red spots are your protein lysates, your antibody binds, you have some detection method, and then you get signal intensities. The advantages of this are that you're looking at protein or protein modification. You can also do a higher throughput screening. It's not as high throughput as oligonucleotide arrays, but you can easily get 4,000 spots on a slide, and depending on how many replicas and dilutions you want to do, and the more of these you do, the more accurate your measurements are going to be. You can easily get 200 samples on a slide, which is nice. And you can also do this with very small amounts of your sample, so you can do precious things like the membrane fraction from hippocampus, and you can also detect low abundance proteins, better actually than you can on a Western blot. So this is just an example. This is phosphor-ERK1-2, and we are looking at hippocampus and cortex from a control mouse and a Down syndrome mouse. These are just blow-ups of these regions here. And what we have done is we have done a 5-point dilution series of our sample, our lysate. This is a blank. And then we have done in this experiment 4 replicates of this 5-point dilution series. So that's 20 measurements of each one of your samples with each one of the antibodies that you screen for. So that's where you can get very good accuracy, and these are just indicating the reproducibility. So the experiments I want to talk about are 3. The results of looking at the TS65DN in its response to learning and memory, when it fails and when it is rescued with Memantine, and then some comparative protein profiling of 4 mouse models, which I'll explain a bit later, and then looking at gender differences in a couple of these mouse models. So the TS65DN and the response to learning and memory with Memantine. So we use the task context fear conditioning. And in this task, you take a mouse from its home cage and you put the mouse into a physically novel cage. So there are attributes of the walls and the floor and the ceiling of the cage that are novel. You let the mouse explore for a few minutes, and then you give it an electric shock and you take it away. If you bring the mouse back sometime later, hours, days, and put the mice back into this novel environment, a mouse that has associated the aversive experience of the electric shock with, the features of this room will freeze. And that is the measure of learning. So when we put the mouse back in, we measure what percentage of its time now it spends frozen, waiting for bad things to happen, and how much time it explores. And this is obviously a good task for the TS65DN, obviously because they fail, right? And this is work from Alberto Costa's lab several years ago now, and this is the time spent freezing when the mice were allowed to explore and then given a shock and then returned to the cage. And the control mice froze about 45% of the time on re-exposure. The TS65DN froze less than 20% of the time. However, when they were given Memantine, they froze at a higher level and at a level that was statistically not different from the level of freezing in the untreated control mice. And then the control for the experience of the electric shock is done in what we call a shock context group of mice. Different group of mice, you put the mouse in the novel cage and immediately give it an electric shock and then allow it to explore. Typically, mice will not learn anything in this because they hadn't associated the environment with the shock. They got a shock and then they learned the environment. And indeed, you see these are the levels of freezing between 10 and 20%. And so there's really no difference in freezing in the TS65DN mice, whether they were asked to learn or in this case, not even asked to learn. The percent freezing is the same. So Memantine is an antagonist of the NMDA receptor, and there are 8 or 9 proteins on chromosome 21 that we would predict would indirectly affect the activity of NMDA receptors. And this is not an easy thing. We can look at these and say, well, you inhibit the phosphatase that dephosphorylates NMDA receptors, and when they're dephosphorylated, they're inactivated. So in the TS65DN mice and in Down syndrome, we would anticipate that the levels of phosphorylation of the NMDA receptor would uniformly be higher. We would also assume, because of other genes, that the dynamics of phosphorylation, dephosphorylation are perturbed. And then there's a number of other things, localization at the membrane. Put all these together, and I wouldn't be able to predict exactly how trisomy would affect the activity of the NMDA receptor or the dynamics, but I would predict it's disturbed. And so the experiment involved control and TS65DN mice. They were 3 months old. We had 8 groups of mice. So our mice that don't learn or aren't asked to learn, mice that are asked to learn, with and without injection of either saline or memantine, for a total of 80 mice. And then we looked at cortex and hippocampus and did some subcellular fractionation. We had a total of 480 samples to look at. So the data I'm going to show you, we've done a number of comparisons. So we define normal learning as the protein profiles, how they change in control mice, because they learn normally. So we compare control mice, the protein expression patterns, in mice that were asked to and successfully learn in fair condition to mice that were not asked to learn. Failed is exactly the same experiment using the TS65DN. They don't learn. And then rescued learning is the TS65DN injected with memantine. And then we also look at memantine treatment. What does it do to the TS65DN or controls? But what does it do to the TS65DN alone? If you just give them memantine, how does that change their protein profiles? And we also talk about baseline, which is looking at mice that were not asked to learn and asking, what does the TS65DN protein expression pattern look like compared to controls, before we even ask them to do something, basically just sitting around. And we do a number of comparisons. But I can't show you all of them, so I'm going to show you little vignettes that I hope will show you, one, the complexity of the data, and two, the importance of the data. So we measure about 100 proteins, and we do this, I should say, 60 minutes after they've been trained. Okay, so we put them in, let them explore, shock them, take them away. 60 minutes later, we sacrifice the mice. And we are looking at a very specific time point, a single time point, but there are prior data in the literature to say that in that 60 minutes, so from sort of 20 minutes to 2 hours, there are a number of molecular changes going on, in particular in the MAP kinase pathway. So that was how we picked this window. Other time points would give us different results. We looked at a whole lot of proteins, everything basically that we could think of that was of interest, that we could get a good antibody for. So they're relevant to learning and memory, or they've been shown to be abnormal in Down syndrome, Alzheimer's disease, or a mouse model of one of these. And we look at MAP kinase, MTOR, as I said, a number of things, and a few chromosome 21 proteins. Very hard to find good chromosome 21 antibodies. And we look at phosphorylated forms and a few methylated, acetylated forms where we can. So in our measurements, a total of this number were detectable in the nuclear, cytosolic, and membrane fractions. And just to give you an overview of this, in each fraction of each brain region, between 30 and 50% of these proteins changed in normal learning, okay? 30 and 50% were different already between the controls and the trisomy mice. And so no matter what you do, a large proportion of these proteins change, okay? So many things are happening in each of our brains as we are standing here, me talking and you listening. Many things are going on in our brains as we are now. So why did the TS65DN fail learning? And this is purely a protein expression answer, okay? So this is the nuclear fraction, male mice, 3 months old, exposed to fear conditioning. So these are histograms that are comparing the level. So black bars are significant differences. These are control mice. Black bars indicate proteins that changed significantly in the control mice after they learned successfully. So this is MAP kinase components. We've got BRAF, phosphoraf, MEK, phosphomec, and so on. Over here, CREB, PCREB, and PCAM kinase 2, and over here we have components of the MTOR pathway. So at 60 minutes post-training and fear conditioning, many things are happening in control mice in the nuclear fraction of hippocampus. Okay? Now, MAP kinase and mTOR, we know these are relevant to learning and memory. We know if you perturb these either genetically or with inhibitors to some of these kinases, you disturb and you can block learning in fear conditioning. So, what do the TS65DN look like? Strikingly different. Okay? If these responses are important for normal learning and memory, when you look at what is not happening or what is going in the wrong direction in the TS65DN, you would guess, if you just looked at these data, assuming this is important, you would guess these mice would not succeed. And indeed they don't. So the abnormal responses that we see in known pathways important to learning and memory, too few, too small, and often going in the wrong direction. What can we do with this? So I want to show you some details of the MAP kinase when we look at a number of different pairwise comparisons. So this is from the previous slide, normal control mice learning, failed learning and memory in the TS65DN. This is what Memantine does to the TS65DN without asking them to learn, just what Memantine does. Not a whole lot. It does give you a spike in phospho-CAMK2, which is probably really important. And it also gives you a spike in PCREB, which might be correcting this decrease. So then if we ask what happens in rescued learning, so we are not seeing in rescued learning the same dynamic responses that we see in normal learning. We're not replicating what we see in normal learning. So yet these mice learn successfully. So the last comparison is showing the baseline, where the TS65DN start from compared to controls. So the TS65DN are starting from a perturbed level of their MAP kinase components. All of these things are elevated already, and many of these are what is supposed to go up in normal successful learning, but they're already high. And so the last comparison we did was we put all these together. What happens if we add up all these different effects? And what we see is that if we add up what happens with Memantine treatment, or what happens at baseline, the differences at baseline, what Memantine causes, and what rescued learning, successful learning causes, then we see that we have replicated almost everything that is going on in normal learning. And so from this, one thing that we now consider is that you need possibly one of two states of a protein. You need a specific level of a protein, and if your brain is already at that level, that's okay. Some proteins. Other proteins, you need actually a change. So in order to propagate a signal, you need the protein level to change. But I think we have both. We have a set of proteins where you need a level, and if you get that to start with, or if Memantine causes it, you're fine. But we still need Memantine to do some things indirectly and cause appropriate changes. That's my thought. And again, the black bars indicate proteins that changed significantly. The white bars were insignificant. And so a last comparison here, this is looking at baseline differences. So again, this is showing you again the MAP kinase, but adding on mTOR. This is all of the differences we saw in these components without asking the mice to learn. But this is comparing the end points in successful learning. So this is comparing the protein levels in the TS65DN after they have learned successfully with the protein levels in control mice that learn successfully. So we see they start off very dysregulated, and through the process of treating them with Memantine and the responses Memantine induces in learning, we end up with profiles that are very, very similar to each other. They're not identical, but they're very similar. And I think it's remarkable. Okay, it's statistics, right? But I think it's remarkable that you can start with this level of perturbation and end up with that level of perturbation. I want to show you a few patterns. So what we want to know is what is really wrong in the TS65DN. You know, is everything that we see that's abnormal wrong and a problem? Or are some of these okay? It's okay to be abnormal in this way. Or is it even good? is that the organism, in some cases, the organism's response to the insult of trisomy, it's now trying to correct this as best it can, and so some of the abnormalities we see are actually compensatory. They're good things. You don't want to target the good things or the irrelevant things. You would like to know what are the problem causers and be able to target those. So I'm going to show you a few patterns, and normal learning, failed learning, rescued learning, baseline, and B-TM is the response to memantine in trisomy mice. So when we look in control mice and look in the Down syndrome mice, we do see some protein responses in the TS65DN that appear to be normal. So this is, the blue bars are always going to be normal learning, the yellow bars are failed learning. And what we see in the cortex nuclear fraction for BRAF is that these levels, failed learning, the mice are responding to the same level that they do in control mice. Similarly, for phosphosarc in the nuclear fraction of hippocampus. So I'm sorry, we have a whole lot of proteins here, and I can't show you all of them, but I'm just showing you a few little examples. So if we then ask what goes on, or where did they start from, in these two cases, the baseline levels were normal, but we see in rescued learning, in this case, that rescued learning is also elevated. BRAF also goes up in rescued learning. What we see over here is that rescued learning, the asterisks mean these are not significant. Nothing is happening in rescued learning, but memantine alone is producing a level of this protein that is apparently adequate for successful learning, because these mice are learning, but is also equivalent to the changes that were induced in normal learning and failed learning. We can have many examples of this. This is Raptor, and again we see that normal, failed, and rescued learning are all producing similar changes. This is histone H3 acetylation at the lysine 9, and they all go down equally. So many responses, and there's many other examples of these proteins, that the responses in failed learning are okay, and they are replicated in either by memantine or in rescued learning. Some normal protein responses appear to be compensated already by the baseline issues. And here are some examples here. Nothing happens in failed learning, and in none of these examples does anything happen in rescued learning or in memantine treatment. So either these are irrelevant or the baseline levels are already adequate for successful learning. And I will say that for interpretation purposes here, we are making the assumption that what we see in normal learning is necessary. Okay, it's potentially not true, but we are making that assumption for ease in pattern identification. So I want to show you what's going on with some chromosome 21 proteins, which I think is really interesting. So again, blue is normal learning, yellow failed, green is rescued, white baseline, and black is memantine. And these are some chromosome 21 proteins, the very famous DYRK1A kinase. Learning goes on in the presence of elevated levels of DYRK1A. Okay, they are elevated at baseline, they increase further when you fail or when it's rescued, and interestingly, they go up significantly in normal learning. Each one of these proteins, I chose these because in normal learning, these proteins are responding. And so intosectin is another protein. ARCAN1 inhibits calcineurin. It plays a role in the dynamic responses of NMDA receptors. Each one of these proteins is responding in normal learning. So chromosome 21, are these genes really playing an important role in normal learning? These data would suggest that we need to think about that they have direct responses to normal learning. And then there's a whole other set of proteins where we see responses in normal learning and nothing happens in failed learning. So these we would consider possibly are contributing directly to the failed learning responses because they are compensated by rescued learning and by directly or indirectly by memantine. So a direct response, correction by memantine, is shown here, and an indirect response is we see a change in rescued learning, but we don't know where it comes from because we know that memantine is not directly changing levels of AKT. So there's a lot of conclusions, and there's a lot of data, and there's a lot of possible interpretations, and we keep looking. We've had these data for over a year, and we keep looking at it and thinking about it differently. So I apologize if these conclusions are not correct. but we do the best we can. So, as I said, we suspect that some proteins are already adequate at a specific level that's abnormal in the Down syndrome mice, and possibly in people with Down syndrome. It's already abnormal, but that's okay because it facilitates learning. Other proteins, we need to change the level, and this may need memantine in order to induce that change. 25% of the responses we see in normal learning are already occurring in failed learning. So not everything is wrong. And we consider these to be necessary, but not sufficient. You want to keep these. And I think it's very interesting that chromosome 21 proteins seem to be directly involved in normal learning. And it's important that learning occurs in the presence of abnormally high levels of chromosome 21 proteins. There are those in the community that think, well, let's find the important genes on 21 and target each one of those with drugs. And so that asks you the question, got 500 genes, how many drugs? But this also tells you that you don't need necessarily to change the levels of individual genes if you can target the pathways that they participate in. Now, I must say that we are very well aware that there are proteins we didn't measure. We looked at a single time point, and we've looked at a single drug. Okay, that's not our goal. We haven't reached our goal. We certainly want to look at other drugs. Do we see the same responses in rescued learning? And if we don't, can we then sort of digest this down, distill it down to a set of responses that are the common ones among multiple different drugs? That's for future funding. So the last two things I want to talk about, hopefully we'll go fast enough. So can we expect drugs that work in the TS65DN to work in people? So far, as I mentioned, clinical trials are not working. So I'm going to show you data where we compared what we see in the TS65DN with the DP10 and the DP17. So these have not been studied much and have never been assayed for drug responses. But what are their baseline abnormalities? If they're just sitting in their cage, how abnormal are they? And then we used a fourth model, which is called TC1. This carries a human chromosome in a mouse background. Okay, there are some challenges with this. It has a few deletions in it. But it was published a few years ago by Lizzie Fisher. So I want to show you a little bit about the complexities. So people are happy with the TS65DN, right? We look in there and then we go to clinical trials. An important feature is, if you look in the DP16, so this is the whole 16 region, it's impaired in the Morris-Water maze and it shows abnormal LTP. Roger Reeves made this little TS1-RHR that is trisomic for the telomeric region of 16, the so-called Down syndrome critical region. And this displays, well, I don't want to go into that. But anyway, if you cross the DP16 with the deletion, so when Roger made when you use Cree-Lox, you get a deletion and a duplication. So Eugene Yu crossed his DP16 with the deletion of this, so basically getting a mouse trisomic for this region, and showed that things were normalized. Super, right? But then he did another experiment, which is really too bad because it made everything more complicated. He crossed the DP16 with the DP17, which are normal in the Morris-Water maze and show enhanced LTP, not inhibited LTP. So he crossed the DP16 with the DP17 and found they were still impaired. Okay, so far so good. He already had shown that if you delete this segment, you rescue things. However, in the presence of the 17 region, if you delete that little red region, you don't fix things, okay? So when you start adding in more genomic segments, you can't assume that what you see is additive, okay? We don't know why, but we know if you take the red region out of the DP16 alone, you correct Morris-Water maze. We know if you take the red region out of the DP16 that also has the DP17 region, you don't correct the Morris-Water maze. So I think that argues that we need to consider at this point in time genes all over the chromosome, and we can't be too happy and self-satisfied that when we see something in the TS65DN, in particular a drug response, that we can fix the same thing in people with Down syndrome. So the red arrows are the 4 mouse models I want to talk to you about, and they're reproduced here. So this is the TC1, a human chromosome. It has 121 chromosome 21 genes, trisomic in it, and then our TS65 with sort of 90, and the DP10 with sort of 40, and the DP17 with sort of 20. So all of these mice were the same age, same sex, and I'm going to show you some data from hippocampus. This is the DP17 for chromosome 21 genes that we measured. None of them, the asterisks, indicate proteins that are not trisomic. So everything is fine. DP17, none of these genes are trisomic. None of them are altered. We look in the TC1. These genes here are trisomic, and they are elevated. Same with these genes here, TAM1 and SOD1. This is DIRK1A and S100 beta. However, this gene, intersectin-1, is not trisomic, but it is also elevated in the TC1. TS65DN, it's trisomic for all of these, but not these 2 here that are down in the 10 region, and most of these are also elevated. When we look in the DP10, save the best for last, only these 2 genes are trisomic in the DP10, and they are elevated, cystatin B and S100, but we see ARCAN1, a phosphatase inhibitor, intersectin-1 again, and we also see APP. APP is the beta amyloid protein encoding gene, mutated in some, or duplicated in some forms of familial Alzheimer's disease. Why are these elevated in the DP10? I don't know the answer. But one must have to assume then that the genes that are trisomic in the DP10 somehow are influencing expression levels of other chromosome 21 proteins. MTOR pathway, this is the TS65DN. Not much happening in the MTOR pathway. Only phospho-MTOR is decreased. DP17, it is perturbed. AKT and phospho P70S6 and phospho S6. TC1 also has some perturbations, again, different, common. Actually, none of these are in common. And then the DP10, again, shows many more abnormalities in protein levels than actually all of the other models combined. No phenotypic features when assayed at 2 months of age in the Morris-Water maze, novel object recognition, LTP, and fear conditioning, okay? Normal at 2 to 4 months of age, but abnormal protein profiles at 8 months of age. So what do we conclude from this? These partial trisomies have partially overlapping, some of them, and some of them are completely non-overlapping. But their baseline protein profile aberrations at 8 months of age are really very different from each other. So we would expect then to see differences in learning and memory and synaptic plasticity. So the DP10 show the most protein abnormalities. These are the least studied, most ignored mouse model. They have no learning and memory deficits assayed at 2 months of age. So what do we have to say? Are all of these abnormalities in MAP kinase, MTOR, Alzheimer's disease-related protein, all of these chromosome 21 proteins, are these irrelevant to learning and memory? I know those proteins are not irrelevant to learning and memory, because if you mutate them in people or mutate them in mice, you cause learning and memory deficits. Yet the constellation of these in the DP10 are not causing learning and memory deficits. Are some of these abnormalities protective? Are there genes that are trisomic in the DP10 region that are protecting or helping to protect from abnormalities induced by other genes? So are there a subset of genes in the DP10 region that are actually protective? Or are there learning and memory deficits at 8 months of age? Okay? Do these mice develop age-related learning and memory deficits? Okay? And importantly, do any of these abnormalities influence drug responses? So these are questions I don't have answers for yet. So very last thing, sort of quickly. We all know most experiments are done with male mice. This is a practical issue. Less expensive in time and resources, and avoids the necessity of controlling for estrous cycle in your females. However, there are data in the literature that are really quite stunning. I mean, I was surprised when I went and looked at this. This is a publication from 2006. They looked at over 100 male and female mice, and they did oligonucleotide arrays. They looked at differences in expression between males and females in a number of tissues. 13% of brain RNAs were different in expression level between males and females, 72% of liver RNA level. And then another example, they mutated the GABA A receptor gamma subunit, and only the females showed elevated levels of inhibition and altered levels of other subunits. Males did not. Now, males may have had other phenotypes, phenotypic features. So there are clear, and you go in the literature, you can find example after example. So TS65DN, 90% of the literature, 95% is done with males. Males are sterile, so you couldn't use them for breeding. Females have small litters, so you keep all your females to keep your colony as big as possible. So there's practical reasons. So we had a group of 8-month-old male and female DP10 and DP17 mice and set about asking with the same proteins. So this is the DP10, the DP17, TC1, and TS65 here. Again, the black bars are significantly different, and these just happen to be AD-related proteins. So when we did the same analysis of female mice, the red arrows indicate what we found abnormal in the female DP10. It is not the same picture at all. And again, if we come back to our MAP kinase and MTOR, here are the male mice perturbations at this age. Here are the females. They don't look the same, okay? We've looked at the females in fear conditioning, and they learn the same as the males do so far. And this is just color-coded, looking at it, showing you a number of other genes and showing you the brain region specificity of this. So this is hippocampus, cerebellum, and cortex. The first column is comparing male controls to female controls, okay? And the second column is comparing trisomy males to control males, and the third is trisomy females to trisomy control females. So we first of all see that blue means down. So the males have a lower level of many of these proteins in hippocampus compared to female controls. Not much difference in cerebellum or in cortex. But we look in the trisomy mice. The hippocampus, as I showed you in the 2 previous slides, the males have a lot of abnormalities and the females don't. So yellow is up. So the male trisomy mice have a lot of yellow. The female are mostly black. No differences. When we look in cerebellum, we see something very different. The males, column 2, largely not much happening. The females, a lot is happening. A lot of upregulated proteins in the female cerebellum. We look in cortex, and not a lot is happening. Okay? That's data. I have no interpretation at this point. So I want to get to the end here then. What we see from this experiment is very clear gender and brain region specific differences, both in the direction and the magnitude of the perturbations. So what happens if we ask, what do drugs do? They may be doing something different in males than they do in females. If any of these proteins are relevant, I think they must be doing. DP10, they encode some interesting genes. PRMT2 is a methylase that activates androgen, estrogen, and thyroid hormone receptors. So is there a gender specificity induced by PRMT2, which is exacerbated in trisomy? S100, I know I talked with people here whose favorite gene is also S100, and there are sex differences that have been noted in S100 expression and in prediction of neuroprotection after stroke, I believe. And TRPM2, TRPM2, sex differences clearly in neuroprotection when you knock this out. I believe that males are not affected, females are. And then we can talk about the DP17 region, which also has sex differences. Not as many as the DP10, but there are clearly sex differences. So the last thought takes us back to drug trials. And this is back to the TS65DN. This is the subset of drugs where they had actually asked about more than one learning and memory test. So some don't, some do. So here again is the name of the drug. This is pentylene tetrazole. These are the GABA A antagonists, memantine again, fluoxetine, Prozac, lithium, and so on. These are the age and the gender of the mice, largely males. And so here's the Morse water maze, novel object recognition, object location, spontaneous alteration, not discrimination, and fear conditioning. And the plus means the drug successfully rescued the deficit. A minus sign means it failed to rescue the deficit, and a plus minus means it improved it, but not to level of controls. And so I think this is interesting data that I have not heard people talk about when they're talking about doing clinical trials with these same drugs, or when they're talking about the results of their data. I think this is important. Some drugs do not rescue all the tasks they're assayed for. For example, this is the Roche drug here. When they looked at this in the TIA65DN, this is from Carmen Martinez-Quay's publication of about a year or so ago, it rescued acquisition in the Morse water maze. So the mice learned better with this drug. But when they took away the platform, it didn't rescue memory. So what does that mean for people with Down syndrome? Other examples are that some drugs could rescue spontaneous alteration and another task, say novel object recognition, or could rescue this and fear conditioning and object location. But other drugs could not. Even though they rescued fear conditioning, they did not rescue spontaneous alteration. Even though they rescued object location, they did not rescue spontaneous alteration. And this one here rescued the Morse water maze, but did not rescue this. And here's another example of fear conditioning in this. So some drugs are rescuing some tasks, and some drugs, those same drugs are not, or rescuing another task. And other drugs, it's the opposite. So clearly these drugs, we don't know, right? We don't know precisely exactly what brain regions are being activated and are required for learning in these, but this suggests that there's something different, that it's not just, one drug fixes everything. So when we go into people where we also are challenged with the genetic variability, are we also being challenged with the wrong task? Are we asking kids with Down syndrome to basically learn something that is using the same types of things as the alternation task? I mean, it's not as simple as that. But if we ask them to do something else, would they be successful? So, last conclusions, pharmacotherapy for ID and Down syndrome. I think this is where we are. And this seems a bit negative, to say that TIA65DN results are misleading. But I think they are misleading, and people are being happy to be misled because they want clinical trials, okay? We all want this. But there are problems with the TIA65DN that we can now address with better mouse models. There are other problems that we could address if we had funding. I mean, I think everybody agrees we want the best mouse model possible. We want to understand how these drugs are working so we can understand if we can expect them to work in people. But in addition to that, I think we need other cell systems, we need other model systems. I don't think we should go from mouse to clinical trials with people without an intervening step where we get some feel for how complex the responses may be in people, and what the genetic heterogeneity does, and how complex the drugs system might be. And so I've just said, future directions. What we really want to do, look at the molecular responses to more drugs, to more learning and memory tasks, and we obviously have to look at gender. We need more better mouse models, but I think we also need to consider what we are assaying and try to develop tasks or use tasks, you probably don't even have to develop them, that assay the functionality of other regions of the brain. And then lastly, this is something I've been struggling with for years, we need human model systems. And there are many ways of getting those. We can culture fetal cortical neurons. I have a collaborator, Jorge Basilio, who does that. ES cells, iPSCs, I talked with some people here. We need to do this if we want to be as efficient as possible when we go to clinical trials. So I stop now. And I just want to mention the people in my lab who have contributed most to the work that I talked about today. Mahi Ahmed has done a lot of the work with the TIA65DN. Aaron Block has done the experiments with the DP10 and DP17. And Ranjita Dhanasekaran has done all of the very large amount of computational work that goes on behind the scenes in analyzing RPA data and doing some of our other bioinformatics work.
Are you aware of the Changing Minds protocol and the Changing Minds Foundation? And looking at the drug list up there, a lot of there are families and groups of people who are using some of those medications with their children, giving them to their children. I noticed the SSRI, the Prozac, the lithium, some of the other medications, and so forth. Are you aware of the Changing Minds Foundation at all?
Yeah, I think so. Okay. Yeah. There's a difference between off-label use and a controlled clinical trial. And I understand that people have, the impetus is different. Scientists control.
You mentioned an intermediate step between really the mouse model and then clinical trials in people. I know that they're actively putting together the biobank now, which will put together tissue samples and blood samples, which hopefully will move us in that direction to be able to find more alternatives. And I also wanted to ask you your thoughts on what's coming with regard to shutting down the extra chromosome and then the restorative kind of medications or processes like, i.e., stem cell research, things of those natures that would help actually restore the brain and the body with regard to Down syndrome.
Yeah, so Jeannie Lawrence's work was really a tour de force where she used the molecular mechanisms that are behind X chromosomal inactivation that occurs naturally in males. And so she used the same molecular constructs and inserted them on one chromosome 21 in a cell line from a person with Down syndrome and showed that she can effectively silence, by the same mechanisms that are naturally working, to silence the second X in men. Tour de force. Can you do this in people? And the answer to that is not now, not simply. Okay, it is a model system in order to study the effects of you manipulating the expression. If this can be done in multiple, I think it would be very interesting to extend this work to multiple individuals. So what are the gene expression and phenotypic consequences of shutting off an extra chromosome in multiple cell lines from multiple individuals with Down syndrome, where the consequence is always the same? I think at the moment the answer to your question is this is a scientific tool. And it's a basic science question, because the challenges with that would be delivering these constructs to every cell in the body. You can do this at the one cell stage at the moment of conception, at which point you don't know this is a Down syndrome fetus. Doing this later on is going to require development of tools that strategically allow you to deliver these. And at the moment, they're not present. Where do you need to deliver them? Do you need to put that in all cells? That's what disomy is. Can you silence a chromosome in only some cells? Could you silence it in bone marrow? I mean, there are a whole lot of questions. At the moment, this is a tool for basic research.
So you use different drugs, like Memantine. So I wonder how long you give the animal for Memantine?
Okay, so so we gave the drug to the animals 15 minutes before training. So 15 minutes. I mean, which by itself I think is remarkable. It was an intraperitoneal injection 15 minutes before, and they learn like normal mice. Other groups have given it to the mice over a series of days, weeks, months, and these drugs have been delivered in water, in milk, in food. Others have been injections. And I think we also have to consider that, and the same drug, different groups have different protocols for drug delivery. And I think we have to also consider that some of these are aversive. I mean, getting picked up and having a needle stuck in you, if you are also already sensitive to aversive events, has its own set of issues, as does using electric shocks, which is also an aversive experience. So every lab with their drug, and many of these drugs have been tested in several labs now, but every lab has its own protocol for delivery, for dose, and for time of delivery.
So, yeah, so actually I wonder if you give the animal the drug like very early, because like early age, because Down syndrome is a neurodevelopmental disease, so you try to affect the development
Sure,
Yeah.
sure.
Another question is like, because your different drug has different effects on the behavior, will it be possible if you combine like a few drugs together and crack all the behavior
Right,
tests?
and we're actually almost funded by NIH to do exactly that with using a very specific computer algorithm that allows you to choose the correct doses. And so we're planning on doing this with 7 different drugs. And what this protocol has been shown, this is work that has been done by an engineer, Chi-Min Ho at UCLA. And he has shown that instead of taking hundreds of thousands or millions of combinations, that using his particular algorithm it's not his algorithm, but his application of this algorithm, you can reduce this to maybe 100 combinations. And what it allows you to do is to use a biological readout, and then the algorithm chooses for you the next best concentrations of drugs. And he's done this for a number, nothing in learning and memory yet, but he's done this for a number of systems. And you find 2 things. One, that it converges to optimal drug combinations and doses very, very quickly, so you don't have to do hundreds of thousands. But the other is that every drug in the combination is at 10 to 100-fold lower than if you use that drug alone. And in all of the cases that he's done, it also gives you better outcome than when you used individual drugs. So drug combinations, I think, is something that a lot of people are thinking of. I think there's a lot of data to say that's what you want to do. And the question is how to do it and how best to develop these combinations. Many of these drugs have a lot of effects that you want to be careful of. So, you know, your other comment was adult versus children. Sure, if you could prevent the development of deficits, it's what you would want to do. But I would also say that there's a large population of adults with Down syndrome that you want to target as well. But the other thing is, I think it's huge that you can take a mouse that's gone through its whole life being unable to learn, and by giving it a drug when it is 4 months or 6 months or 10, 12 months of age, and have it learn normally. That says something about the brain that we had not been anticipating. And the other thing is, the third point here is that approval for drug testing, especially of some of these neuroactive drugs in children, is a challenge, right? Now, there is a group that has done Prozac, fluoxetine in pregnant dams, TS65DN, and shown that the pups born when they're aged to 3 months or 4 months learn normally, okay? And so from this comes the suggestion that we should give Prozac to women who are pregnant with a Down syndrome fetus. There are negative consequences to SSRIs on the developing brain. So I think there are a lot of issues when you start talking about manipulating using drugs in fetus or in young children. There are a lot of things we don't know.
Yes, I noticed that in your lecture you touched on the use of folate in your, as for treatment in infants, and the results were good. Over the years, I've heard that folic acid, I think, what is that, B6, given to mothers when they're pregnant, that's high risk as far as Down syndrome works. And I've also heard over the years that the occurrence of Downs, but I guess this is old news, happens more often in women over a certain age. I think women over 35, when the mother's over 35, than younger women. And so over the years, I've also heard that they gave B6 and B12, because you have to have B12 to balance the B6. You know, one neutralizes the other. Well, B vitamins in general, folate and B6, I think, are the same. Am I right? Okay. Folic acid, okay, to pregnant women, and this greatly reduces the chance of Down syndrome. But this is the first time I've ever heard giving B to infant children, and it actually, reduces it actually has a positive effect. I know this is very complicated with the extra chromosome, and I was going to ask the question the lady asked, is there some way to neutralize that third chromosome? And you said, well, maybe in rats, but not in people.
Not even in rats or mice.
Right.
It's in a cell
okay.
now, in a tissue culture dish.
Yeah, I was thinking instead of neutralizing it, like making it more to where it looks like the second chromosome on the same, on 21, replicating it and making it like identical rather than trying to change it. Okay, I think, or get rid of it. I think that's more realistic because, well, in time when we figure it out, okay, we'll be able to change or alter the chromosomes, but we can't eliminate one for another unless that's done, you know, well, like she said, stem cells or something like that. You know, we can't just get rid of a chromosome in a rat or a person just by zapping it out. Okay.
Yeah, so the trials that were done with folate were actually done with a logical, based on logic. There are 8 or 9 genes on chromosome 21 that function in the folate pathway, folate transport, and downstream 1-carbon metabolism. So there was a very good rationale. And these genes are not trisomic in the TS65DN. They're in the 10 region and the 17 region. So you couldn't do the same kind of folate trial and expect the same outcome in the TS65DN. So that was a very interesting trial, and they've kept a fairly low profile. They've been very cautious in their interpretation, and I thought it was quite exciting, but they said, no, we need to do a bigger trial. What to do? So the whole focus of the thought in my lab is that you can't get rid of the extra chromosome, so you want to compensate for it. And it's where you compensate for it. Jeannie Lawrence did it by turning off the genes on that chromosome. Our thought is you go downstream and you integrate the effects of all of these trisomic genes and you find key things that need to be regulated. And that's the focus of these drug treatments.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.